Hi, hello to all the AP and Telangana JL DL aspirants. Welcome to Chandu Biology Classes. In this session, we are going to discuss about male gametophyte. Clear? So yes, in this, initially we are going to discuss about anther structure, microsporogenesis and development of male gametophyte. We all know the male reproductive structure of an angiosperm is stamens, right, which has filament and anther. Anther of most of the angiosperms is two-lobed. It is two-lobed. The microscopic view, you can see it clearly, it is two-lobed and each has two thecas. So, it is called as dithecous. It is called as dithecous. When only one lobe is present in the anther, it is called as monothecous. In only few of the angiosperms you can see monothecous anther. The dithecous anther, in dithecous anther you can see 4 microsporangia. You can see clearly 4 sporogenous tissues where microsporangia are present. Clear? And here the anther is diploid. Sporogenous tissue is diploid. All the cells present in the sporogenous tissue are diploid, which are called as pollen mother cells or megaspore mother cells. In monothecous anther, if you observe, each anther consists of only 2 microsporangia. In dithecous anther, you can clearly see 4 microsporangia. And then if you look into each sporogenous tissue has some specialized cells called as Archesporial cells. Archesporial cells initially divide and form primary parietal cells and primary sporogenous cells. Primary parietal cells will undergo anticlinal divisions and they form the wall layers of tapetum. They form the wall layers of anther. They form the wall layers of anther. As such, after primary parietal cells undergoing anticlinal divisions, you can clearly see endothecium, middle cell layer and tapetum. Primary parietal cells will give rise to secondary parietal cells whereby they further divide and give rise to tapetum. And then primary sporogenous cells, they will divide and they form pollen mother cells, which after meiosis they form microspores. Pollen mother cells will form microspores. This is the division of the specialized cells in the sporogenous tissue, which are archesporial cells. Now let us once look into the theory also. Archesporial cells, by the time anther primordia becomes 4-lobed, certain specialized cells get differentiated below the epidermis in each lobe. These cells are larger in size when compared to the neighboring cells. Each cell will possess dense cytoplasm and a diploid nucleus where it has complete set of chromosomes. These cells are known as archesporial cells, which is also called as archesporium. Archesporial cells can be arranged as into one vertical row or in few plants like 2 vertical rows and in few plants like 3 vertical rows. As such, archesporial cells arranged only in one vertical row, you can see Dionia, Sansevieria and Impatiens. Okay? And if Archesporium arranged in 2 vertical rows, you can see Boerevia and Erythroxylum. Archesporial cells arranged in 3 vertical rows, you can clearly see Ophiophogone and Cephadesa. We have to remember examples, friends. All the examples wherein archesporial cells are arranged as 1 row, 2 rows and 3 rows and their examples have to be in our mind. And then if you look into whatever divisions we have observed, like periclinal divisions of archesporial cells will form outer primary parietal and primary sporogenous. Primary parietal will undergo both periclinal and anticlinal divisions and they will form the anther wall. Primary sporogenous tissue will undergo few mitotic divisions and they form primary sporogenous tissue. Okay? So, here you can clearly see, here both anticlinal and periclinal divisions takes place. Both anticlinal and periclinal. Both anticlinal and periclinal divisions takes place, and that is how wall layers of anther are formed. And sporogenous tissue purely undergoes mitotic divisions and they form microspore mother cells. This microspore mother cell undergo reduction division and they form microspores. And then if you see, yes, 4 types of anther wall development is recognized so far. What is the 4 types? Basic type, dicotyledonous type, monocotyledonous type and reduced type. In basic type, if you see, primary parietal layer will divide and they gives rise to outer secondary parietal and inner secondary parietal layer. Outer secondary parietal layer, primary parietal layer will give rise to outer secondary parietal layer and inner secondary parietal layer, where outer secondary parietal layer will give rise to endothecium and middle layers, and inner secondary layer will give rise to anther inner middle layers and tapetum, anther inner middle layers and tapetum. This type of development is observed in few families. This basic type of development is observed in few families like Anacardiaceae, Lecetidaceae and Nyctangiaceae, Tiliaceae and Winteraceae. And then if you look into dicotyledonous, monocotyledonous and reduced type, only slight changes are observed in middle layer. Middle layer here in dicot type is formed directly from the outer secondary parietal layer, and middle layer in monocotyledonous type is formed from inner secondary parietal layer. Reduced type is wherein middle layer is totally absent. And then wall layers, if you see, we have 3 wall layers. Those are epidermis, endothecium and middle layers and tapetum. Total 4 wall layers are present. So epidermis, if you see, this is the outermost wall layer made up of several cells in a single layer that shows only anticlinal divisions. And endothecium, if you see, they help in dehiscence of anther and they develop fibrous thickenings by the pollen grains. These endothecium fibrous thickenings are usually alpha cellulosic fibrous thickenings, and they are usually absent in those flowers which are completely closed till fertilization, like cleistogamous flowers which are observed in viola, common pansy, and like as such cleistogamous flowers where bud pollination is clearly seen. Flowers will never open at all, and certain cases cutinization and lignification can be seen. In the maturation, when microspores mature to pollen grains, you can see the maturation of endothecium where cutin and lignified depositions are observed in the endothecium. That is why it is called as hygroscopic in nature. It is hygroscopic in nature. As microspores mature to pollen grains, they get dehydrated so that they can dehiscence easily and they can release out the pollen grains. And then if you see, middle layers are clearly visible during the early stages of anther development, like 3 to 4 middle layers are present in holoptelia, and among these layers except outermost, the rest 3 will degenerate. In ranunculus, 2 middle layers are formed. The inner one degenerates while the outer one still persists. Inner one will degenerate and the outer one will still persist. Absence of middle layers was noticed in the smallest angiosperms like Wolffia and Vallisneria, which is a hydrophytic plant. Next, if you see tapetum, it is the innermost layer of the anther wall and it surrounds the microsporogenous tissue. One or more divisions may occur in the tapetal nucleus during first meiotic phase, and tapetal cells may have one or more nucleus and they will show nuclear fusion, and they also contain polyploid nucleus. In tapetum, the nucleus is more than one per cell. So you can observe polyploid nucleus in the tapetum, and we have two types of tapetum. One is secretory or glandular tapetum, second is amoeboid or plasmodial tapetum. Secretory or glandular tapetum, these tapetal cells will degenerate totally by the time meiosis is completed, and vacuoles develop inside the cytoplasm. These are the signs of nuclear degeneration, and these cells are useful in nutrition of microspores especially. When it comes to amoeboid or plasmodial tapetum, in this type of tapetum, cell walls of tapetal cells will break completely, and due to the fusion of the protoplast, dense cytoplasm is formed which enters into the locule. This is called as amoeboid tapetum, that is shapeless. Okay, as cell walls break and protoplast fusion happens, this enters into the locules and they get shapeless. That is why it is called as amoeboid tapetum. This tapetum closely associates with microspore mother cells, and hence it is called as periplasmodium. Okay, that is why it is called as periplasmodium. And major important function is Ubish bodies, these are present in this tapetum, are helpful in formation of pollen walls. Hence the chemical substance, chemical constituents like carotenoid esters of Ubish bodies is similar to that of sporopollenin of the exine of pollen grains. Okay, sporopollenin of exine of pollen grains are formed from the Ubish bodies itself, we have to remember. And these Ubish bodies are in what type of tapetum? Amoeboid or plasmodium tapetum. In the next class, we will see different types of microspore developments like successive type, simultaneous type, and different types of tetrads as well. So stay tuned, and I would like to end up this class by giving you a fantastic information that here in Narayanguda in Chandu Biology Classes, we have started offline classes for JL, DL aspirants of both AP and Telangana. Every new batches commences 15 days. Okay, new batches commence every 15 days. So what are you waiting for? Join Chandu Biology Classes today itself, which is in Narayanguda, Hyderabad. Thank you.